Good morning, everyone, and welcome to today's presentation. I'm Mr. Shailesh K. Verma, and I'll be guiding you through this session. We'll be focusing on the WIDAL test using the tube method, which is an important diagnostic tool. In this presentation, I'll cover the basics of the test, how it's performed, and its clinical significance. So let's get started and explore this topic together. So on this slide, we're looking at a serological test used to diagnose typhoid fever. Essentially, this test detects the presence of antibodies in the blood that are produced in response to salmonella typhi and paratyphi infections. These are the bacteria responsible for causing typhoid. The test was developed quite a long time ago, in 1896, by Georges Fernand WIDAL, which is why it's called the WIDAL test. When we ask, what is the WIDAL test? We're really talking about a method that helps us identify whether someone has been exposed to these bacteria by checking for specific antibodies. It's a significant tool in the diagnostic process, especially in areas where typhoid is common. This test forms part of the broader theme of how we detect and confirm infections through serological methods, which are crucial for timely and accurate diagnosis. Now, let's take a closer look at the causative organisms behind these illnesses. First, we have salmonella typhi. This bacteria is responsible for causing typhoid fever, which is generally more severe and can lead to serious complications if not treated properly. It spreads mainly through contaminated food and water, so maintaining good sanitation and hygiene is crucial in preventing its spread. Next, we have salmonella paratyphi, which causes paratyphoid fever. This illness tends to be milder compared to typhoid, but it's still important to recognize and control to prevent outbreaks. Both of these bacteria highlight the importance of understanding how different strains of salmonella can cause varying levels of illness, and they remind us why hygiene and safe food practices are vital in controlling these diseases. Now let's look at how the WIDAL test works. When we take the patient's serum and mix it with specific antigens, such as O, H, AH, and BH, we are essentially testing for the presence of certain antibodies. If those antibodies are present in the serum, they will react with the corresponding antigens. This reaction causes visible agglutination, which means the particles clump together. That clumping is the key indicator that the test is positive. So, in simple terms, the principle of the WIDAL test is based on detecting these specific antibody-antigen interactions. If we see agglutination, it suggests that the patient has been exposed to the pathogen and has produced antibodies against it. This is how the test helps us diagnose infections like typhoid fever. Now let's take a closer look at the materials we need for performing the WIDAL test. First, we have the patient's serum, which contains the antibodies we're going to detect. Next, we need the WIDAL test tubes, and in this case, we'll be using 24 of them to run multiple tests or controls. We'll also require specific antigens, O, H, AH, and BH. These are the key components that help us identify the presence of typhoid antibodies. Normal saline is important to dilute or prepare the reagents and samples properly. To handle small volumes accurately, we'll use a micropipette with tips, ensuring precision in our procedures. And finally, the water bath is essential to incubate the test tubes at the correct temperature, typically around 37 degrees Celsius, to facilitate the antigen-antibody reactions. All these components are integral to ensuring the test's accuracy and reliability, and they connect to the overall theme of precise diagnostic testing in microbiology. Let's start by taking 6 tubes and adding 4,000 microliters of saline to each one. This will serve as our base for the serial dilutions. The next step is to add 210 microliters of serum into the first tube. This creates our initial 1:20 dilution, which is essential for accurate measurements down the line. From there, we transfer 2,100 microliters from the first tube into the second tube, which already contains saline. This transfer effectively dilutes the serum further, moving us from a 1:20 to a 1:40 concentration. We continue this process sequentially, transferring larger volumes each time, specifically 2,100 microliters, to prepare subsequent dilutions, ending up with a series from 1:20 all the way to 640. This serial dilution method is a fundamental technique in our experiments because it allows us to create a range of concentrations. That way, we can accurately determine the serum's activity across different dilutions, which is critical for reliable analysis. Let's take a closer look at the tube arrangement on this slide. We have a total of 24 tubes, organized into 4 rows and 6 columns. Each row represents a different antigen. The first row is for the O antigen, which is crucial for identifying the bacterial serotype. The second row is for the H antigen, related to the bacterial flagella. Moving to the third row, we have the AH antigen, and in the fourth row, the BH antigen. These additional antigens can help differentiate closely related strains. This setup allows us to perform multiple tests simultaneously, making it easier to determine the specific serotype of the bacteria we're studying. By understanding this arrangement, we can see how serotyping becomes more efficient and accurate, which is fundamental in microbiology and infectious disease diagnosis. All right, let's walk through the final steps of the procedure. First, you'll add 500 microliters of antigen to each of the respective rows. This ensures that the antigen is evenly distributed and ready for the next steps. Next, you'll perform a serial dilution by adding 500 microliters vertically, which helps us determine the highest positive titer accurately. After setting up the dilutions, incubate the plates overnight in a water bath. This allows enough time for the antigen-antibody interactions to occur properly. Finally, the next day, observe the plates carefully for agglutination. The key here is to note the highest dilution level where agglutination still occurs. That's your highest positive titer. This process is crucial because it helps us quantify the antibody levels in the sample, tying back to our overall goal of understanding immune responses. Let's go over the example report. The O antigen is positive up to a 180 dilution, which indicates the level of its presence in the sample. Similarly, the H antigen is positive up to a 160 dilution, showing a slightly higher concentration or detectability. The report also notes the presence of AH and BH antigens as observed, which helps in identifying specific bacterial strains. This kind of detailed information is crucial because it guides us in understanding the bacterial serotype involved. Remember, the higher the titer, the more confident we are about the antigen's presence. Overall, these results help us pinpoint the exact strain and guide appropriate treatment or further testing. It's a key part of our diagnostic process, tying into the larger goal of accurately identifying and managing infections. For academic use only. Thank you for your attention today. I appreciate your time and engagement throughout this presentation. Remember, the key points we've discussed are essential for understanding the broader context of our topic. If you have any questions or need further clarification, feel free to reach out. Again, thank you for being part of this learning experience.